Dr. Yin, there's been a lot of research I know that BGI have been doing recently about ALS. Why are you doing this?
Actually, ALS is a name maybe for common people. They just know the name. But how about the cause? Which genes, which factors can cause the ALS? Maybe we are not very clear. ALS is not a single disease. It's a family. I think at least dozens of, even hundreds of, the similar, the symposium we can call ALS. For example, the Hawking is ALS, and Tele is also ALS, but totally different. So the first important thing we must know how about the genetic or genomics background with the ALS family. For example, if the mother is normal, the father is normal, but one of their child that they may be very young, they just became an ALS patient. So we must know the mutations, even the novel mutations. Maybe it's not only for therapy, but it's really helpful for how to prevention and prediction. On the other hand, we are trying to how to deal with the ALS. We use our latest technology we call the Omics. That means Spatiotemporal Omics. Thank you for a lot of very brief the ALS patients. They donate their muscle tissue to us, and we cut the muscle tissue into a very, very thin layers. Each layer is only 10 micrometer. That means the single cell layer. And with our technology, we can see with the genes, there's something wrong, there's some mistake, there's some harmful mutations in it. We found some very interesting markers. For example, IL-17B. In the past, the researchers only find the IL-17B in blood, but not in muscle cell. So we want to block it. Just like this year, the Nobel Prize, the microRNA, we synthesize the ASO, that means the antisense oligo. It can block the IL-17B, and we do the trials on mouse. Already today, it's around 5 weeks. Hopefully, we can get the positive result. So maybe we can uncover some answers to the ALS patients. On the other hand, we found a very interesting discovery. If we are healthy people, we cannot find this type, the muscle cells. But in ALS patients, some muscle cells, they're abnormal. And we can see some disorders, not only genomics, but also protein and metabolics. And we can see same region, if there's some IL-17B, the abnormal muscle cell is also here. They are in the near region. They are very close. Maybe one caused another.
Are you hopeful of finding or helping scientists to find a cure for ALS within your lifetime?
Yeah, I hope so. Maybe not all subtypes I can solve, but I think several subtypes, there already some lighting is here. For example, the most clear subtype is we call the SOD1. SOD is a very, very common. Even we use cosmetic, we use SOD. SOD1 it can cause subtype of ALS. Now it's we can use the gene therapy or similar treatment ways we can deal with this subtype. And other subtypes, I know a lot of the drugs or therapy solutions is already undergoing. Only for me or me with Tele, maybe we are not enough. But a lot of researchers, other research teams, they are embarrassed by the Tele spread. So I think if you want to go fast, just go alone. If you want to go further, go together. Just like 5 years ago, we have not powerful enough to deal with the Mendelian disease, but now we have gene editing. So I think time by time, step by step, we can solve the most diseases already exist in human society. But humans still belongs only one small portion to the nature. We must have our fear and respect to the nature. and bear in mind the human nature. I think it's balance because the Earth can exist without a human being, but a human being cannot exist without the Earth. Just put in mind not only the science, the technology, and also the love, the ethics, the civilization. That I think that's the really harmony, common future for not only human beings but also the whole planet.